Okay, good morning, everyone. I'm truly delighted to be back in Ireland and to have the opportunity to present my results of my project at RCSI. So today I will be sharing key findings from my research and my work on microRNAs and their potential role in MS. So microRNAs are small non-coding RNAs that can regulate the expression of different genes at post-transcriptional level. The same microRNA can regulate the expression of different target genes, and the same target gene can be regulated by different microRNAs. Specifically in MS, they have been studied as potential biomarkers, since their levels have been related to different MRI parameters or the use of treatments. They could also provide new insights into disease pathology, since they have been involved in immune cell function or remyelination. And last, the therapeutic potential of the inhibition or overexpression of microRNAs has been already explored with promising results. So during my thesis in Spain, we explored the presence of different microRNAs in cerebrospinal fluid from MS patients, and we found higher levels of miR-21 in those patients that have gadolinium-enhancing lesions in their MRI. There was also a moderate correlation between miR-21 levels and neurofilament levels in the CSF of these patients. And regarding clinical parameters, there are correlations between miR-21 levels in cerebrospinal fluid and the number of T2 lesions in MRIs and basal EDSS. So during the PhD, I came to Ireland and I spent 3 months in the lab of Dr. Claire McCoy at RCSI, and that time we established a preliminary experiment with brain slices to study microRNA profiles during demyelination and remyelination. And we could find some different microRNAs that were upregulated, the blue circles, and other microRNAs that were downregulated, the red circles. But interestingly, we found that miR-21 was upregulated in both demyelination and remyelination comparing to control samples. So after these results, Claire and I decided to apply for different postdoctoral grants to try to continue this line. And we got one project that was microRNAs as therapeutics for neurorepair in multiple sclerosis, whose main research goal was to try to combine human data with experimental models to try to identify microRNAs that could be involved in the damage and repair associated with MS. So we established murine organotypic grain slices where you can induce demyelination using lysolecithin, and you can examine demyelination and remyelination ex vivo. In addition, using immunofluorescent staining, you can overlap myelin basic protein and neurofilament, and that way you can assess the extent of myelination in the culture. We also wanted to perform single-cell sequencing analysis in this model, and we capture individual cells using the BD Rhapsody platform, and later on we use Nigen scarf web to analyze the data generated in the experiment. And last, we also established different murine in vitro cultures, and we did some activation with lipopolysaccharide. So first of all, we tried to replicate what I had done before during my predoctoral stay. So we increased the number of brain slices setups, and in this screening phase in where we use open array panels to screen microRNAs, we could corroborate that miR-21 was still upregulated during demyelination and remyelination compared to control conditions. And when we tried to validate these results in a posterior phase with a higher number of experiments, we could find that this upregulation was higher during demyelination. So we decided to perform single-cell sequencing analysis because we wanted to identify gene expression signatures in specific cell types in the model during these stages. And the first analysis revealed the existence of 24 different clusters according to specific expression markers. These 24 clusters could be grouped in 7 bigger clusters, since some of them belong to the same cell type. So we could find astrocytes, microglia, neurons, and also cells that belong to oligodendrocyte lineage. Functional analysis within these clusters reveal transition from proliferating and differentiation to mature and reactive oligodendrocytes. Also saw astrocytes that could promote repair, but later on produce inhibitory glial scars, and also saw different microglia phenotypes that might contribute to myelin debris clearance or to inflammatory damage. We also saw that there were cell population frequencies differences among the experimental conditions. For instance, we found that astrocytes presented higher levels of cells in myelination, and specifically from one of these clusters, and that neurons, for example, presented lower levels at the end of the culture. We use public repositories to try to determine the potential origin of miR-21 in the central nervous system, and using microRNA tissue atlas, we could confirm that although it's not the main organ, miR-21 is expressed in the brain. And specifically, analyzing the CNS microRNA profiles generated by Hoy and colleagues, we could determine that microglia was the cell type that most expressed miR-21 within the CNS. So from now on, we try to focus on microglia, since it was the potential origin of miR-21 in the brain. And in the single-cell sequencing data, we analyzed potential dysregulation of genes, and we found that some miR-21 target genes, such as LCN2, CCR1, AHR, and IL1RN, were dysregulated in the brain slices. Specifically, the 4 genes were more expressed during myelination, that was when miR-21 was downregulated, and IL1RN presented lower levels during remyelination after 7 days in the culture, and the other 3 genes expressions presented the opposite tendency. Interestingly, these 4 genes have been previously related with MS in different studies. During the project, I also spent 3 months at Queen's with Yvonne Dombrowski, and there we established different primary in vitro cultures. And first, as we were interested in microglia now, we established primary microglia, and we activate with LPS, and we could see that miR-21 was upregulated in microglia, but it was not in mixed glia cultures. So we made a first approach to try to inhibit miR-21 in microglia. So to conclude, our results could suggest that miR-21 can have a potential role in active lesions of patients, but also in demyelination and remyelination. And some of miR-21 target genes have been found dysregulated, which could open new therapeutic avenues. So now we are focused on trying to implement microglia-specific manipulations to try to enhance remyelination in multiple sclerosis. So to do so, we are trying to optimize manipulation of these candidate genes that we have described previously in human microglia models, and we are trying to optimize brain slices with the endogenous microglia reduced to try to co-culture these human microglia with the specific manipulations with the brain slices to see if we can enhance neural repair. So before I finish, I would like to introduce to you the Spanish MS Research Network that I recently joined. It's formed by 21 groups, and we are open to collaborate. So in case any of you are interested in collaborate with us, I would be more than happy, or have more additional information, I would be more than happy to answer to all of you. So you have my email here, and thank you to all of you, to the organizer, for having me the opportunity to be here. Thank you so much.